As you know, this is the IDCRC Mentoring Lecture Series. We're back again this month with another opportunity to learn and to ask questions and to grow. And we are very lucky today to have Dr. Elizabeth Brown, a professor at the Vaccine and Infectious Disease Division at Fred Hutch, and the Statistical and Data Science, the director of the Statistical and Data Science Unit in the Infectious Disease Clinical Research Consortium. She is a biostatistician who's been engaged in the design and analysis of ID trials, including COVID-19, our favorite, HIV, AIDS, human papillomavirus, is the principal investigator for the Statistical and Data Management Center for the Microbicide Trial Network, and also is the director of the Infectious Disease Clinical Consortium Statistical and Data Science Unit. She is coordinating all of this information nationally and internationally, and is a force to be reckoned with us. And we are really excited to have her share her knowledge with us and for you to be able to ask her questions too. So I'm going to turn it over to her because she's much more interesting on this topic than I would be.
Thank you. Okay, let me try to share. Is that working?
Yes. Yes. You make it full screen.
There we go.
There you go.
Okay. So today we're going to be mentoring each other. And you might be wondering how you're going to mentor me. So I typically, when I work with people, it's more of a one-on-one. Here's how our relationship is going to go. Well, not exactly like that, but here's how we're going to be working together. Or let me be honest, more typically it goes, Elizabeth, let me tell you this really detailed thing about this pathway that regulates blah, 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 that you have absolutely no possibility of understanding because your last biology course was AP Bio in 11th grade. We're just going to be honest there. And so then it goes through how do we go from this real detailed information that investigator wants to give me to really the big picture information that I need to understand how to best design this study and how to move forward with the statistical questions. So it usually comes on a case-by-case basis because everybody comes to me with different levels of experience working with statisticians and kind of understanding these potential roadblocks, or really, they're not really roadblocks, but like bumps in the road that can slow down progress. So this is the first time I've tried to like put together some ideas about working with a statistician that are more general in nature. And I found a paper that really influenced most of this talk, and I'll make sure it gets sent around later. So my thought is to just kind of go through some lists of ideas and hopefully be very discussion oriented and be more responsive to your questions than to me just lecturing at you. So I'm not, hopefully I can see all of you if people put their little hand raising signal up or even just want to interrupt me at any time. I welcome that very much. Otherwise, I think we'll all get tired and go to sleep and take our afternoon naps, as I like to call it, my 5-minute afternoon nap that COVID, not just COVID work time, seems to require now. Okay, but before we go, I'm not the only person at the SDSU that some of you might interact with if we're talking about specifically IDCRC analyses. I'm the PI of the subaward that funds the SDSU. And right now we have 4 other faculty statisticians, and I expect this to grow because the work is growing, who work with me on the statistical aspects of the IDCRC work. That's Holly James, Barb Richardson, Olivier Hirian, and Clara Dominguez Islas. And you may have met some of these folks already through various IDCRC trials that you may be involved with. And we also have an epidemiologist working with us who's actually Barb and Jen are both at UW. So Jen Balkus, who a lot of you may know, she worked with the STI network that was blended with the VTUs to form the IDCRC. But we're mainly housed at the Fred Hutch. So what I love about being home, there are some weird background home noises going on right now that are a little distracting. I'm going to try to ignore them. Sounds like my son might be missing, but Okay, so first I'll just go through some of these ideas about what's the role of the statistician in developing your proposal. And when I put this together, I'm kind of in the world of secondary analyses. So we already have some kind of interesting large trial that finished or large study that finished, and we want to figure out how to answer some other questions that might be of interest from the data collected in that trial. And this is very, happens a lot. I remember the first trial I was ever involved with was as the protocol statistician was HPTN 024, which was a study of antibiotics to prevent chorioamnionitis, which then in turn, the hope was that preventing chorioamnionitis would prevent mother-to-child transmission of HIV. The odds ratio at the end of that study was 1.01. So completely null finding. It doesn't get much more null than that. But we still published about 25 manuscripts from that study because of all the data that was collected and the value in that data in that population. So I think there's a lot of hope and anticipation that we'll be seeing some similar opportunities coming from, for example, the CoVPN studies, especially the data that comes from the CoVPN sites, or maybe some of the other trials that we'll be doing as part of the IDCRC. So what is my role or one of my colleagues' roles when we're working with you? And I kind of put these in the order that I feel like it's maybe not as necessarily clear to people that our role exists in. A lot of times people will come to me with a proposal and it will be like linear regression of X on Y. And the last thing I want to see is in a proposal at that stage is what the investigator thinks the statistical analysis should be. The main thing I want to know is what is your hypothesis, right? So the first step I want to have in working with an investigator is clarifying their research question or their hypothesis. So, and then making sure they're clearly stated, they're adequate, they're realistic, and that somebody can translate them into a statistical question. So, or a model or a statistical test that can answer that hypothesis. So that is number one. The second is really identifying what is the outcome variable you're looking at, and are the endpoints clearly defined. So this is a place that can be tricky too, and can also sometimes require some discussion because sometimes it's not as clear as a single endpoint like an infection. If it's, say, viral load, people have an idea that they want maybe the maximum value observed or an area under the curve, and those all have different kind of properties and pros and cons with what we can do with them based on how the data is collected and what the correct statistical approaches are. So that's really the second thing to make sure you've discussed with your statistician. And I've had this happen to me before where I feel like we're clear on the endpoints and we've written the statistical analysis plan, and then someone will go and change the endpoints thinking that doesn't really matter, or they'll add endpoints or move them from secondary to primary. And that really has implications throughout your proposal. So throughout the process, it's really important to keep your statistician in the loop on your endpoint discussions. Okay, and I think that the rest are the things that people expect to work with their statistician on. And so to make this clear, I guess, I'd say, your statistician is someone you collaborate with, right? And I think that's, and actually, your statistician can be a mentor to you too. But I think that's an issue sometimes that can come up is people tend to think of a statistician as someone that you just say, calculate this p-value for me, tell me this power. But it's somebody who should be involved in collaborating in your science and really a part of your team and working with you. And so I think, and maybe this third part isn't as obvious then if you're not in the mindset of thinking about the statistician as your collaborator, and that's to ensure you have a study design that can appropriately and adequately address your proposed hypotheses. And then I think these other ones are pretty probably pretty obvious to most people, and that's this proposing methods that and statistical methods and really other methods to ensure bias, blinding, and stratification is handled properly in the study, to review assumptions used for the sample size estimation, and defining a clearly specified appropriate data analysis plan. And maybe one thing that's missing here is really ensuring that you're studying the correct population. And especially in a secondary analysis, when you're starting with this fixed population that you can't control, are you including the right subset of the population into your analysis? So these are really, I think, like the main things that when you first meet with your statistician, the order in which you'll really need to work through these problems, and sort of the first, I think honestly, the first thing people think they want to talk about is what's the fifth point on here, which is sample size. They come straight in, I just need a sample size, I need a power test. And it's like, no, actually we need to start right back up here at the top with your questions and your hypotheses, and make sure we're both on the same page and that they make sense, and that you'll have the data collected that can adequately answer them. So I do want to pause here just in case anybody has any questions or discussion here. I don't have a lot of slides because I really want to make sure I answer questions if they come up. But if not, I can move on to the next list.
I wanted to say something. You know, I think that your point about always making sure that you're continually having the conversation as you're developing your objectives and aims and the outcomes, because I think that that's something that oftentimes we reorganize things and we think about how we're going to do things, and it kind of comes to us and we're like, oh, we'll just flip this around. And understanding that that has broad implications is something that we don't always realize at first. And I think that that's probably one of the most important things that I heard you say. And because I've done that wrong, and you only have to make that mistake once before your statistician wants to kill you, and twice if you want them to leave you. So it's really, really important lesson to learn, that everything that we do is so intertwined, that we really are a team working together to help to design things in the best way possible.
I think that the team thing is so important. And I'll be honest, that's not usually a problem I find with younger investigators. I feel like younger investigators, and maybe this is changing for more experienced or mature investigators as well, but really embrace and understand the role of the statistician, and how it's really a collaborative relationship. I think it's been a long time. I can just remember from early days having a investigator once come and say, I want you to test these 10 variables, all of them with these 10 outcomes, which gives you like 100 p-values. And I said to him, what's your hypothesis? Like, I want to design the right analysis. What are you hypothesizing here? And I was probably a year out of grad school. I was an assistant professor, and he didn't answer me. And he immediately called someone who he thought could get me fired because, you know, he felt like that how I was overstepping my role to ask him what his hypotheses were. And it's funny because years later, I would still see senior people who were involved in that whole mess, and they would say, oh, I can't believe you had to put up with, I'm omitting the name, I'm so sorry you had to deal with that, et cetera, et cetera. But I haven't dealt with anything like that in quite a while. So, but yeah, I think it's really key. The things that come up are like that. People just aren't thinking, they're trying to get their work done quickly and efficiently, because we all have so many pressures on us. And before you may know it, you've written your statistician into a hole that they can't climb out of, or an analysis they can't design to fix what was put in a protocol or a proposal. So yeah. Any other questions so far? Things to highlight? I think maybe the next slide might help more with some questions, which I don't know how to fast forward. There we go. Yeah, so this is really, again, reiterating this. And I think a lot of this is putting this in different ways to be very specific about when you come into a meeting, things that we're going to need to know. So the information you need to have together. And again, I want to go back to this example because it's hard to pull examples because they're about specific people. And even if I don't say the names of the people, I don't want to, you know, say something negative about a group I work with or something. But I do have this role with the CFAR where I help junior investigators. They have to have a statistical consult before they can submit their applications here at the UW and Fred Hutch. And some of them honestly will just come to me and start telling me about this, the example I gave at the beginning, like some regulatory pathway or something like that, and how this type of cell and that type of, I don't even know, because I don't know these things. And I'm like, that's interesting, but what is it you want to learn? And is this a lab study? Like, are we going to be doing a study in people? Or are we going to be doing a study that's tightly controlled in a lab where, you know, the specimen may have come from a person, but everything else is so highly controlled, it's no longer like a population level experiment. So that's one thing to the first thing to talk about. Who is your population? And even if it's some work you're doing with a phase 3 study, and you say, for example, my population are the people enrolled in the Moderna phase 3 trial. Is it really all the people? Is that really your inclusion exclusion criteria? You know, who is really becoming part of your sample? Again, your hypotheses, very important. Outcomes of interest, how they'll be measured, how frequently they'll be measured. Is that really, you know, some of the things we discussed then is that really frequently enough? Like a current example is thinking about SARS-CoV-2 infection as opposed to disease, and how frequently that has to be measured because it's a transient infection, right? It's not like measuring HIV where I could measure it infrequently and get a pretty good estimate of when somebody became infected. If I don't measure frequently enough, and with SARS-CoV-2, I may miss it altogether. And so how might that impact then any kind of comparisons I want to do between groups? If for example, my hypothesis is that it, you know, an intervention shortens viral load, but I'm only going to measure viral load once every 2 weeks. But I'm going to hypothesize that its effect is through that duration shortening, but I'm picking up maybe something different, a combination of incidence and duration shortening. The last, I don't think we talked about this in a randomized trial. The intervention is pretty obvious. It's what we've randomized somebody to, but in a secondary study, it may be some other variable collected throughout the trial. And we need to be sure that we've identified that carefully, and then think about appropriate comparison and control groups. So I think really when you come to meet, and I think a lot of groups have templates for secondary analyses, and I should have grabbed one that we use in the MTN that really gets to all these things. And I don't even allow a space for statistical analyses in that unless somebody has a statistician already on their team, because that, as I said before, people will often come and they won't put anything except this linear regression model they're going to fit, or this Cox proportional hazards model they're going to fit, and not even tell you what the primary parameter of interest is. So that's for your statistician to figure out. This right here is really what your statistician needs to know so they can get to the next steps. So I will pause again because I do want to be conversational, but maybe if I get through these first 3 slides, you might see where some missing pieces are that you might have questions specifically about. I feel like there is something missing. There is a slide missing. That is, I wonder if I accidentally deleted a slide I didn't mean to. So let me just go through this real quick. Okay, I actually know what it is I want to say. So one thing I want to make sure I talk about is I think that is on a later slide. So sorry about that. So to get to some other tips, and again, this is like an iterative process. When you meet with your statistician, it's likely going to take more than one meeting. And again, always consult if you're planning on changing aims, objectives, endpoints, the ordering of doing something, how you're going to select samples. And you bring the scientific area expertise, the curiosity, the enthusiasm. So let your statistician bring the statistical expertise to complement all of that you're bringing. That is my one big tip that I think we've hit on multiple times today. So I did have some other specifics, and I think the one I want to focus on is really the sample size, because I think that's the other, it's power and sample size, and I'm putting these together. And I think for secondary analyses, this is a big issue. And it's a little different issue, right? Because if I'm designing a new study from scratch, I might say, how many people do I need? I can go through sort of the standard sample size calculation. I want this power, I want this alpha, I'm measuring this outcome. Here's what I know about it already in people who don't have the intervention. Here's what I assume the effect size will be. When I'm doing a power or sample size calculation for an existing data set, and really doing this for a completely different reason, which is to say, is this wasted time to do this analysis or to do further sampling? Is there, given this limited number of people I have, can I either estimate something with enough precision so that maybe it's a new measure that we don't have on the subsample, so we want enough precision around it to get a meaningful estimate of it? Or if I'm comparing two groups, is the difference between the two groups that I would be able to detect based on the power I want, the sample size I have, the distribution of sample size between the two groups or more groups, will I have enough data at the end of the day to make a meaningful statement on the difference? So I could give an example where recently we looked at a study where somebody was wanting to look at a measure in the blood, and we asked for the sample size calculation or the power they would have, and they sent it back to us, and the effect size was there, but the units were missing. So it was hard to understand what it meant. And when we kind of looked up more information about this measure, the effect size difference that they would have been able to detect from doing this study was larger than the range that value actually takes on in the general population. So it was like, I mean, at the end of the day, we decided that knowing this value was so important in this group of people that we were fine with that. But that's an example of where it's really important when you come into these sample size calculations for these secondary analyses that it's a little more nuanced, because it's really about convincing people that you can get an answer that's meaningful with the data that you have available. So that could either be projecting a confidence interval width or an effect size. So you have to, when you're coming into these, you're going to have still these same requirements that you would typically have. But now, instead of having the study tell you about sample size, you might be coming in with that sample size, and you might be coming in with the power and looking for the effect size that you can detect. Or it would be, I think, more rare to come in with a sample size and effect size and say what kind of power you have, but really to look at what kind of meaningful effect size you can detect in that data. The other way that might be useful is if you're doing more sampling. For example, you have an intensive lab procedure you want to do, you might find that you don't actually need to do it on the whole cohort of people. You could actually go for a smaller, more targeted cohort of people, because your sample size is so large that you would be overpowered if you did this analysis or extra testing in everybody. So I think it's kind of a key thing when thinking about secondary analyses, that you're really asking different questions about power and sample size than you might typically be asking in a study where you have more, in a newly designed study where you have so much control over what that study design is going to look like, whereas in the secondary analysis, you just have to take advantage of what's already there, and you can't really add to it, change your population, change the distribution of your of your exposure. It's already fixed for you, and you just have to take what's there. And then there are designs that can help with that. So I'm going to go backwards if I can figure it out. Yep. And that's, for example, I think the most common type of secondary analyses you might encounter for a study where you're interested in doing more lab measures is the case control or case cohort kind of design, where you take all cases and then have some sort of targeted sampling of non-cases. And this is really common when new and expensive exploratory assays are being proposed. And these are incredibly complex to design, or they can be, and to calculate your power and sample size around. But again, these are the things that your statistician you work with can help you figure out. If you want to know something like this from this large group of people, how to hone in on a smaller group that you actually can afford to do this experiment on. So I do want to pause because I feel like I could just start going on about all the different things that float into my head, and that's not going to be super helpful to you guys. So I want to pause and see if there's questions, because I think we've covered the main aspect of what your initial meeting with the statistician might be like as you're building this collaborative relationship. So I'm hoping there's some questions out there. Helen promised me questions. So Helen, I'm going to call on you if you are here. Erin, thank you.
Hi, thank you so much for this presentation. I have been fortunate to work with statisticians before, so I wish I'd had this before I started interacting with them. I do a lot of I've done different types of research, but I generally tend to do basic immunology. And I think one of the things that I have a hard time with is that a lot of times I'm doing something very new. So there's nothing out there on it. And it's the same with mice. And everyone always asks me, how many do you need? And my answer is, well, we don't know because we've never looked at this before. So I think that's the thing that I have a challenge with, because once I get that preliminary data, then I can tell you how much variation there is in the method with that group. And then that can help me then design if I want to do a larger study. But I feel like with a lot of basic immunology, it's like, well, we're limited by what we can get, or we're limited by what we can ask for. So do you have any advice?
Yeah, so I'm going to give you my perspective that's not probably shared by all statisticians, and maybe not a lot of statisticians. In that specific case, in that kind of case you're describing, I think it's fine to come in and say, I have this many animals that I can sacrifice, and that's it. And it's not, to me, then, how do I put this? I feel like in a lot of lab studies, when your sample size gets really small, I just don't even sometimes see a role for statistics, right? The signal is there or it's not, and it's so highly controlled that all these sources of variability that we're worried about when we're designing these large population level studies really aren't there in these highly controlled lab studies. That being said, you still have to convince people that you're going to get enough information to be that it's worth investing their money in your project, right? So I think if I was doing that, I'd be sort of looking at variability, or I would probably do something on the standard deviation scale. So that's sort of a trick that we often use when you don't know what the distribution of an outcome will look like, is to be able to say, well, if it's one standard deviation of difference or two standard deviations of difference, that's when it will become, that's when we'll have 80% power or something like that. So I sort of take the actual scale of the outcome out of it and just generalize it to standard deviations. And that seems to be acceptable to most people. But I'd be curious, when you've done this, how do you usually approach it?
Honestly, the past few studies I've done, which have been human subject studies or animal studies, they're less happy with this, but I've said it's a pilot study, and I've said we're gonna look at a small number of people, and we're gonna get some understanding of, is this immune response we're looking for, is it even there, right?
Yeah.
And if it is there, then we can start to ask other questions, but sometimes that's all we're asking. So we usually start with, very small group sizes. And we know that we may not be statistically powered, but like I said, if we're starting from zero, it's very hard to, make assumptions, because immune responses to vaccines and infectious diseases can be so highly variable. I could make a guess, and anyway, so that's good to know.
I am very sympathetic. That's like the whole world of COVID. The first COVID study I did a year ago was a household PEP study. And what do you expect the probability that the household member will get infected is? Like, what proportion of household members do you expect to get infected? Like, nobody knows. You just have to make something up and talk about, and in that case, we just had to include plans to revisit the sample size. And luckily we actually were pretty spot on in terms of household transmission. So
nice.
It didn't turn out to be a problem. But have you ever gotten pushback on your approach of just saying this is pilot data? I would think that's acceptable, but
I haven't from the IRB. They understand that we're kind of like going out into, unknown waters. And most of the studies that I've done have not been with a lot of high risk. So it's been acceptable. But I find with animal studies that I've had to like pull it from some other paper on some totally different pathogen or vaccine or something, and they don't really like me to just say I don't know, and we're going to start off with a small number. They want me to justify it somehow, and I understand that. I mean, I do. I think it's I think human and animal research is tightly regulated, but it's been that I find is my hardest challenge, because then once I have some understanding of what's happening, any kind of understanding, then I feel like yes, then I can do interesting predictions with that data. But until I have anything, I find it's hard. So
it's really hard. And even in population level studies of things that you think might be well understood, it can still be really hard to find the preliminary data that you need, or maybe someone has it but they didn't publish the parameter you need to know from it. Like if you're doing repeated measures data, you might need to know the correlation coefficient, and that's typically not published. Although it's funny that I say that now, because somebody did actually ask us in a paper review recently to include it, and I wonder if it was their own frustration that they haven't been able to find it for this, for data in the past. But so it seems like, I think I imagine that people on the call have a wide variety of areas that they're putting proposals in on, ranging from lab data where you're coming up with the data for the first time, like you have to create that preliminary data, versus maybe thinking about doing case control, case cohort studies. So I'm curious, is there anybody on the call who's done a case control or case cohort proposal? Maybe you're lucky not to have, or is thinking of doing something like that, or anybody who's thinking about studies that are coming from some of these like CoVPN vaccine studies. Nobody's interested in secondary. And that's what I keep hearing, is that all the mentees want to do some work with the CoVPN studies.
Yeah, I'm getting involved in at least one of these, the PEP study in particular, looking at the subgenomic RNA versus CT values. And it's kind of like, I don't know I think I can phrase some of the questions reasonably well because I have some familiarity with the type of tests they're doing, but whether or not, I think we're going to run into is it clinically significant versus statistically significant is what I'm thinking we're going to run into a lot. But all of this, I know better how to design something from scratch than I do how to go back in and reanalyze and re-justify the choices. So I look forward to working with you on this.
Yeah, so Megan and I are working on this together from the HCQ PEP, well, the treatment study too, that I didn't mention the treatment study earlier, but I did mention the PEP study. I'll say that's the hardest part of a secondary analysis is working within the confines of a study that wasn't designed to answer your question. And so you run into, say, missing data that has, doesn't impact the primary analysis, but impacts you. The subgenomic RNA is interesting because that's also still very experimental and I think still a little contentious. And I don't want to get into too many specific details on that, but it's that same kind of idea though, that you didn't design the study for that. So there's some samples the lab will tell you, we can't run this on this sample, and yet we ran it on another sample that was just like this one and got a value. So what do you do with that? And those are like biases that I could probably spend like 40 minutes right now talking to Megan about that because I've seen some of this data already, but I am empathetic, and I think that's why, and Megan's already heard me say a lot of this because she has to sit and listen to me on these HCQ calls where I'm like, we all have to talk about this. We have to talk, and here's my opinion. And they've all been very subjected to my opinions so far. But I think it's good. And but that's another example of where it's going to be really important to start talking to the statistician right away. And that's an even tougher one because the data is already collected. You have no control over what happened with that. The subgenomic RNA is there, the regular RNA. I don't know. What do you call it when it's not subgenomic?
Whole RNA? I just said CT value.
Yeah, I know. Right. So that'll be fun. But it's also the hard part about that is how do you make sure that you've defined all your analyses a priori when all the data is sitting there begging you to look at it, and then and sending you down pathways that are not really part of your primary mission, let's say, but your curiosity is overwhelming you. And so it's very easy to get distracted when you have too much data, I think, for the statistician and for the team. And Helen, I think you know about that. I'm call on Helen now because I've been working with Helen on a paper recently.
And I think we're in version 3, 3 different analyses now.
And I didn't help with that. So just to let you know, your statistician isn't perfect when I had this idea for this plot that I still love, but it completely sent us off track, I think.
Yeah, I think, I don't know, as a junior investigator also, I got so excited, but having that guidance from the biostatistician or something more senior to, it's like, we can't really answer so many questions. The message won't be clear. So just trying to get back on track and be more focused. It was very helpful from working with you, Elizabeth. And I think that also that separation of like our clinical thoughts, but what it's statistically possible to answer, or it's something meaningful that we can answer with the analysis, and it's also important to kind of like get, we have many ideas, but can we really test that? Can we get something meaningful out of that? Those are very helpful input from you.
Yeah, and I think that's thinking about can we really test that? I mean, that's something that we encountered, I think, with the paper we worked on, which will hopefully be submitted soon. So that's also from the HCQ PEP study, which is not an IDCRC study, but trying to describe so we had daily PCR testing on people exposed in a household for 14 days. And so trying to describe these viral trajectories that we saw in these participants. And I think, people came in with ideas they had from previous studies, not in SARS-CoV-2, I think from like herpes shedding studies, and where you have thousands of people versus the less than 100 people that we had in this study, and trying to figure out like no, we can't divide this into 10 subsets and describe averages over time, because that's so few people. So what are some other approaches we can do to kind of get you these answers? So we took some modeling approaches in that case, which I think it's turned out really well. I'm excited about it. That got to a similar thing, but recognizing that when you have less than 100 people, you can't, and you only have 14 days, you can't do full expected trajectories for each duration of shedding that might happen. So yeah, I'm excited for that to come out, Helen.
Soon.
All these like secondary analyses are such tough papers. So many little things to juggle, and little questions to, when do you stop worrying about bias, or when do you just accept that you couldn't do everything perfectly the way you would have wanted to if you could have designed the study yourself from scratch. Like if you could have done if you had designed a study that was a modeling viral load trajectories, you would have probably done 28 straight days of testing if possible to really characterize everything from short to long. You would have ideally been able to get antibody tests at baseline to see if they were previously, or at the tail end of an infection instead of just getting an incident infection. But you don't have that, and I think it's helping to understand what you can do and how you can interpret your data in the face of these limitations that often come up in these secondary analyses. So thank you for letting me pick on you, Helen.
Yep. I have a question with the vaccine trial, I guess secondary analysis project. Is that something that I know you always think loud and you have great ideas and it's like that could be a project. So how does it work? Are these I guess main investigators come in with these secondary analyses or different sub-projects in coming to you, or are these things generated and up there for somebody to take on and work on, or I'm not sure if there's any structure on that.
That's a great question, and I wish Kathy could have been on the call today. I think she got called away for all this stuff going on with J&J. So that I couldn't, it's so confusing because these studies, and not even those, but you could also think like the Regeneron study. Some sites, I think a couple of the VTU sites were involved with the Regeneron PEP study. So what would be the process in these companies / NIAID funded collaborations, co-VPN collaborations? It's really unclear to me how that's going to work. I do think that anything you could, and this can't really quote me even though this is getting recorded, if it comes from data from the co-VPN sites, I think that is up for grabs, but the larger data set may not be. But what the mechanism will be for that, I'm not sure that's been determined yet. And I think that's still up in the air. And how that would work, I think, I like to think, and I hope that we have enough availability of our statisticians at the SDSU to always be available to you guys if you need help. I know that Clara recently helped, and I'm sorry, I'm forgetting who it was, a junior investigator at Emory put together a grant. Okay, it was you, Erin? Yeah, so I hope that was a positive experience, and I know she was happy to do it.
Was. I just wrote her today, actually. So we got feedback from my R21, which was scored. I don't know if it was fundable because unfortunately I had this great idea and 2 people published in Nature and Science Immunology after I submitted it. So it was a good idea.
It was a good idea.
Yeah, but anyway, so the good news is that I think they really liked one of the aims. The other one, they were just like, it's great, it's very significant, it's just not novel now. So I think what we can do is collect preliminary data, but then I think I just need to come up with another mechanistic aim, and I think we'll be okay. And then Act 1 gave us samples. So I actually wrote to her because we did a power analysis based on this preliminary data in a bioarchive paper, and we were guessing we needed a ton of samples, but they've since publish papers with much smaller numbers of samples showing statistical significance. So I'm hoping we can kind of pare down. So instead of getting 4,000 tubes, hopefully we can get fewer. Because I have a very intricate analysis, like immunological analysis to do. So it's not really one of those things where it's not like a PCR test where I can just run the, you know what I mean, through a machine.
Oh,
But
yeah.
anyway, Clara was great. And yes, I will, when I speak to her, if she answers my email, I will tell her that you gave a great presentation. And now I know if we communicate how to like approach, I mean, we did talk about the hypotheses and everything. She was great. But I feel like I know better now because I feel like I'm one of those nerdy scientists who tends to jump into like, this is a really exciting question I have, and you know, I want to look in these populations and stuff. So anyway, it's been helpful.
Well, you were not my example at all when I gave that example. Nobody on this call, I can guarantee you, was in any of my examples. Mostly I tried to pull from my CFAR experience. Which I shouldn't put the CFAR people under the bus either, but yeah.
I find it's very, oh, sorry, I didn't mean to.
No, go ahead.
I find it's challenging to communicate with statisticians, and I'm glad to have this guidance. And I also find, although it's been very productive, actually Claire was really great. I've communicated with other biostatisticians before where I feel like I had to kind of start from like, it took a lot of background for me to explain. And I felt like I wasn't doing a good job, but I was trying my very best. Because I don't understand what they're saying because I have zero biostatistical training, which is unfortunate, but then it is really challenging. And then I find the same challenges when I go to people who are doing what is it called? Computational biology. It's exactly the same thing for me. So they're talking about all this like Bayesian and all this kind of stuff. And I'm like, sorry, what? And and you guys understand that. And I'm already like admitted how unintelligent I am by saying that, but like I get very quickly confused. And I used to be pretty good at math when I was like in college and grad school, but now I feel like talking to them about these things, I also feel like that's very challenging. Like those two interfaces I have a lot of challenges with because there's different languages and yeah, anyway.
It's funny because there are things that we do like a statistician and a computational biologist or a data scientist or, you know, the different names people go by. There's procedures we do that are exactly the same and we call them completely different things. But it's funny thinking about listening to you talk, I was just reminded of this big confusion we had putting a different proposal together where the statisticians were using the word analysis and the lab folks were using the word analysis. And we're talking about two different things, right? Because you have what you call an analysis within the lab. That is not a statistical analysis. It's the whole procedure of getting some summary information from your sample, right? That we would then use in our statistical analysis. And it took me a while to figure out what our disconnect was because I was like, these timelines make no sense. Like, when are we getting the data so we can do our analysis? And they're talking about the timeline for doing their lab analysis. And when it just felt like I had such an epiphany when I had this realization. I don't know how in like 20 plus years this has never happened to me before, but it hasn't. And I was like, oh, you know what? We're using the same word. We have to use, I have to say statistical analysis and not just analysis. And yeah, it was really funny and we got it all worked out. Although I still will say analysis and they're like, well, we'll have that to you. And I'm like, you're not, who's doing the analysis? Oh, wait, yeah. It's just funny. So, but yeah, I mean, and that's the other thing too about your statistician. We do want to hear about your science. I'll be honest. We can't just dive into the middle of it, into the deep end of it. Just like you don't want me to start talking about priors for a Bayesian model without giving you any context. You know, as a statistician, we work in so many different sciences too. So I've worked in cardiology, cancer, infectious disease. I'm sure there's stuff I've done I'm not even remembering right now. Observational studies, controlled randomized trials, lab studies. We're all over the place and we try to do the best that we can. But as I said, I literally haven't taken biology since 11th grade. Although I did take at graduate school, they had this class that was awesome, physiology for epidemiologists. So I took that, and that was great too. But really, I have like, I'm learning as much about the science as I can as I go, as I encounter different projects. And I think all of us are like that, and biostatisticians. I think, you know, I realize too that I'm giving a lot of perspective from a more senior level biostatistician, and it's going to be really a different experience working with a junior level biostatistician, which some of you will have to do, and they won't be as confident in telling you that you're going down the wrong path. So hopefully, if you're working with a junior level biostatistician, they have their own senior level person mentoring them, like Clara does from behind the scenes, who can check in, and they are able to get that kind of guidance from, like what I wasn't getting when I was dealing with this gentleman who I mentioned earlier, who thought he could get me fired because I asked him to give me a hypothesis. So, you know, we're all different. And I think that's why it's kind of hard to give a single talk that's going to tell everybody how to collaborate with another person, when that each of those, you know, partnerships have different people with different needs and different ways that they work together, come from different places, different amounts of knowledge about biostatistics or about infectious disease or this specific pathogen or the host response to that pathogen. So, you know, there's probably a lecture somebody could give to biostatisticians about how to better work with their scientific colleagues as well. But I am remembered, I don't know if you've ever seen it, and I'll have to find this link and have it sent out with this paper I mentioned. This cartoon that came out probably like 10 or 20 years ago, one of those little stick figure cartoons about somebody collaborating with a statistician and asking what their sample size has to be, and it keeps going back and forth. But I only have 2 people. What's my power? But then what's my saying? I'll have to find it. It's really hilarious, and probably I should have started with that. Not that I think any of you would do it, but it is pretty funny. So
Yeah, I don't. Oh, sorry. Go ahead. I don't know that you can ever know all the biology out there. Like, I'm in a niche myself. I think a lot of the MDs that I work with, because I am an immunologist. I mean, immunology is like cancer. I mean, I don't know all immunology, right? And I feel like I've gotten that reputation. But it's like I have a niche, and I can try to move on that a little bit. But it's like if you're used to working with, you know, lung cancer or ovarian cancer, that doesn't mean you don't know everything about, you know, diffuse large B-cell lymphoma.
Yeah.
But so I think it's challenging. I think it's more helpful for us as a biologist to understand the basic terminology. I really think that would be the most helpful personally. But that way, at least maybe we can meet you halfway. But like I said, some of my initial conversations with biostatisticians were like, you know, what's the variation in your method? I'm like, well, I've analyzed a couple samples to establish it. So I don't, you know what I mean? I don't really know.
Yeah.
So.
Well, and those are the kind of things that if you have to put sample size calculations in, you can always do a range and say we think it's between this and this, and here's what the power is across that range, and how much the true value of this is really going to impact my power. I mean, power and sample size calculations, it's an art as well as a science, right? There's so many unknowns and so many assumptions you have to make, and so many ways your study might produce data that is inconsistent with your assumptions. You know, I can think of examples where this even happens in phase 3 studies, where someone makes an underlying assumption about an incidence rate and an effect size, and then does the interim monitoring for that trial, and it's so inconsistent that you end up having to stop early when you probably should have just designed your study differently to begin with if you had known that better. But by stopping early, you've kind of hurt yourself in ways that you don't expect, that I don't want to get into today, because that's a completely different topic. But yeah, it's kind of an art of just hitting that fine point and thinking about mitigation strategies if your assumptions are incorrect. Like, is there a way you can go partially through your study and recheck your assumptions so that if you're way off base and now there's absolutely no way that you'll get a meaningful result, either, you know, statistically significant or a clinically meaningful effect size, that you can stop and not waste your resources and put your attention to other endeavors that might be more helpful. I will say I can share these slides. I'll probably, I don't know how to move through them, clean them up a little bit more, but I did try to put some things in here that are kind of helpful guidance about things, including, like, you know, variables and type of data that you'll be collecting. I think the most challenging one to think about, and I'm glad it's here, are these like derived variables. And I think, you know, in immunology, this is something, or your lab studies, that will come up a lot. It's not really just that observed point, but it might be some summary of a curve that comes out, or some summary of a count of things above a threshold. And so the sources of error and variability are complex in those kind of analyses, and they can have implications on statistical approaches that you pick. I gave an example earlier about picking the maximum observed value. So peak viral load is an example where we all know that the max has unique characteristics and is a little bit biased because you're more likely to pick an outlier, right, in only one direction. So how do you want to account for that? And maybe it doesn't matter, maybe it does matter, versus an estimated max by fitting a curve like a model and taking out the maximum from that model versus the observed maximum that it's going to have a better error distribution possibly. So yeah, I think, you know, it's good to think about if you're lucky, you're in the case where you just have a 2-level exposure and a 2-level outcome and you randomized and you're done. Really simple analyses. Our CTs are simple. Analyzing randomized data is easy. We don't have to worry about biases and how we can account for those and what other variables we need to include, which I guess is another point that I didn't really get into here too much, but that will be the other question, is really in these observational studies or secondary analyses, which are essentially observational studies, what are your potential confounders and how did you define those? So I recently reviewed a paper, for example, where they just came up with a list of 10 confounders. And I think they had about 5 or 6 different outcomes they wanted to look at. And they just used the same confounders for every analysis, and they had 90 participants, 90 subjects in this. So now you have a regression that always has 10 variables with 90 outcomes. It's just way overspecified and no information about how they picked these confounders. Was there some sort of previous studies where these were confounders, or some kind of hypothesized biologic rationale for these things to be confounders? So, you know, that's kind of in the paper phase of what you would expect to be showing, but you need to go in with your idea about what these confounders are, and likely your confounders are going to run into a lot of missing data. So what are you going to do? Because it's not part of the study design to collect a lot of these variables. They're just conveniently there. Yeah, I think those are other things to think about. Is there another question? I actually don't know how long we're supposed to go.
I think we're at the hour.
Oh, wow. I didn't even notice. You guys were so much fun. I thought I was going to look down and be like 12:20 or something.
Yeah, no,
Yeah,
it's great. I think everyone really appreciates everything that you've done, and we may ask you to come back to do either a reprise so that we can bring new questions, or an extension, because I think that there's a lot of interest in the topic and how we can engage with our, you know, together as developing that team. So I want to thank you very much, Dr. Brown,
thank you
for your for your time and for your efforts. And I know that the rest of the group will be very happy to have you as a resource. And I think we'll have to figure out the best way, from maybe some feedback from the team here, how we can best engage you to utilize your knowledge to help this group grow.
I'm happy to help. Thank you so much, and good luck to all of you.
Thanks, everybody. Have a good day.
Thank you.
Thank you.